Indonesia is a country with the highest number of fracture cases. A survey done in 2013 by the Health Department of Indonesia found that 25% of fracture cases lead to death, 45% were physically disabled, and 15% were psychologically stressed. Fractures in humans and animals are generally caused by different traumas such as being hit by a hard object, being hit by a vehicle, and falling from a high place. Therefore, the need for materials for fracture healing is increasing, as it is important to properly treat fractures in animals as well as humans. Based on the problems mentioned, we as a team are interested in conducting a literature study entitled Bonegraft Application with a Combination of PRP and CHA Biomaterials from Cow Bone Waste in Animal Fracture Models. This is an innovation and a new method in extracting CHA biomaterials from bone waste and combining it with PRP, which has never been carried out by previous studies. It is hoped that using a combination of CHA and PRP can increase the use of high-cost bone implants. The tools used include petri dish, crucibles, porcelain plates, pestle and mortar, hammer, 150 mesh sieve, analytical balance, muffle furnace, characterization using XRF or X-ray fluorescence, pan-analytical type epsilon 3, and XRD X-ray diffraction, pan-analytical type Expert Pro, and mouse cage. The materials used include cow bone waste from Malang City Slaughterhouse, male Whistler rats aged over 2 months with an average weight of 150 milligrams, and feed and drinks for cats. The tools needed in making histological specimens are scalpel blade, tweezers, filters, automatic processor machines, vacuum machines, blocking machines, freezer with a temperature of -20 degrees Celsius, microtome machines, microtome knives, 40 degrees Celsius water baths, object glass, cover glass, special rack for coloring, and oven with 60 degrees Celsius. The material for making histological preparations is in the form of pieces of bone tissue that have been fixated with 10% neutral formalin buffer. The solutions needed are absolute ethanol, xylol, paraffin, glycerin, 99.5% albumin, hematoxylin solution, lithium carbonate, eosin solution, DPX, and decalcification solution. This research was an experimental study using a total of 30 rats, which were divided into 5 treatment groups, 1 negative control, which are healthy rats without treatment, and 4 controls with therapeutic treatment. Therapy 1 is for mice with fractures and CHA paste with a concentration of 25% then bandaged. Therapy 2, in which the fractured mice were given 50% CHA paste and then bandaged. Therapy 3 is rats with fractures and then given 75% CHA paste then bandaged. Therapy 4 is rats with fractures that are given 90% CHA paste and then bandaged. Bone waste from cows are washed using running water, which are then sun-dried. Next, the bones are crushed through pounding with a hammer and mashed using a bone crusher. The fine powder is sieved through a 150 mesh sieve to separate small particles, then analyzed by XRD. The cow bone waste is then calcined at 500 degrees Celsius using a muffle furnace for 4 hours to produce calcium carbonate compounds. That powder obtained as a result of calcination is added with platelet-rich plasma. Finally, PRP is mixed into CHA powder in the form of a paste. The right tibial bone of the mouse is used as a place for the defect. In an anesthetized state, the area of the operation is shaved and given 10% betadine. This is then covered with a hollow duct. The location of the surgery is on the medial side of the right tibial bone, close to the joint between the tibia and femur, around 1 cm from the joint, making a straight horizontal incision around 1.5 cm. The flap is opened to reveal the tibia bone. Drilling was performed using a round type burr with a diameter of around 3 mm, with a defect length of around 3 mm. By doing this, it will penetrate the cortical bone, which has a thickness of around 1 mm, and the spongious bone. In the process of drilling, the area needs to be wet, and this is done through giving 0.9% of natrium chloride. This is to prevent necrosis or death of surrounding tissue cells while the individual is still alive. 5 groups of mice that has been made into animal models of caudal limb fracture, in a condition where the bones are still exposed, were given CHA paste therapy with a concentration of 25%, 50%, 75%, and 90%. then the outer side is closed again with a bandage to fix the fracture so that the healing occurs in the correct position. After the fracture was made, the wound of the positive control group was immediately closed and fixed with a bandage on the other side. The outer bandage is changed periodically during the 21-day cure period. On the 3rd week after the fracture treatment, the mice were euthanized by means of cervical dislocation. After being decapitated, a 1.5 centimeter bone block was taken by cutting the tibial bone of the mouse using a bone burr in the directions of 0.5 centimeter to medial and 0.5 centimeter to distal of the observation area. The sample was put into a buffered formalin solution and labeled according to the therapeutic treatment. When a fracture occurs, it will cause hematoma to happen, which is when an injury causes blood to collect and pool under the skin. Next, an inflammatory response will occur. Since bone fracture is an injury, it incites an inflammatory response. Inflammatory signals result in a local increase in macrophages, which in turn releases factors and cytokines that promote healing. As a result, granulation tissue will be formed. Endochondral formation occurs between the fracture ends and beyond the periosteal sites in this tissue. These areas are less stable, so the cartilaginous tissue forms a soft callus, giving the fracture more stability. Finally, bone formation happens, leading to bone remodeling.